So our last step, stage 4 here. So this is our final stage of cellular respiration, and this is the big one. This is where we're going to finally get ATP. We're going to make a lot of ATP here. So this one is going to occur in the inner membrane, across the inner membrane. So if we were to zoom in, we would see, of course, what the inner membrane looks like. It has your lipids, your protein, some steroids, not steroids, sorry, cholesterol here, which used to make steroids. And this is going to be huge in our last step. So this is where the last step is going to occur, across the inner membrane. All right, so the last step is called, or last stage, sorry, is called oxidative phosphorylation. Oxidative phosphorylation, when you look at the word phosphorylation, remember phosphorylation means adding a phosphate onto a molecule. And in this case, we want to add a phosphate onto an ADP molecule in order to make it ATP. Now, in the previous steps that we made ATP, remember we made it in glycolysis, and we just saw that we make ATP in the Krebs cycle as well. In both of those steps, that was substrate level phosphorylation. We took a phosphate off of another molecule and put it onto ADP that made ATP. Here, that's not going to happen. We're not going to directly take a phosphate from another molecule, put it onto ATP in order to make our ATP. I'm sorry, put it onto ADP in order to make our ATP. So with oxidative phosphorylation, there's a different process to add a phosphate onto ADP. So there's two parts to oxidative phosphorylation. The first is the electron transport chain. Electron transport chain is a series of redox reactions where you're going to reduce and oxidize electron carriers. Okay, so the electron transport chain is passing on electrons, which of course we know is oxidation and reduction happening. Okay. The process of this, of passing these electrons along in the electron transport chain, is going to slowly release energy and also allow a hydrogen gradient to form. We're actually going to be able to push hydrogen ions out of the cell, or not, sorry, out of the cell, out of the mitochondrial matrix into the intermembrane space, and that's going to be huge for the second part of oxidative phosphorylation. So the second part is called chemiosmosis. Chemiosmosis sounds like this big scary word, doesn't have to be. So chemiosmosis, all it is using the movement of the hydrogen ions back into the mitochondrial matrix, and that's going to basically provide energy to make your ATP. So chemiosmosis is just the movement of those hydrogen ions back in, and that's going to drive the production of ATP from ADP and inorganic phosphate. So the electron transport chain, if we look here, we see that we're going to remove electrons by pulling off that hydrogen there and separating the electron from the hydrogen atom. And we're going to pass those electrons along a series of protein complexes here. These littler circles here are simply proteins. Almost all of them are proteins. But these proteins are special because they're electron carriers, so they can easily accept electrons. As you go down the electron chain here, so as we go down this way, each electron carrier is a little bit more electronegative. So that means it has a stronger affinity. It wants to grab those electrons stronger than the previous. So as you go all the way down here, we see at the very end, one of the most powerful ones here, the most electronegative, is going to be oxygen. So that's what's going to drive the force. It's going to pull the electrons down. So they're just going to keep passing the electrons from one to the other, each one being a little bit more electronegative and wanting to pull the electron away. So as the electrons get passed down, this is slowly going to release energy, and you might be thinking, yes, we're finally going to make ATP. As we release the energy and you drop the electron down this electron transport chain, and we're finally going to make ATP. However, that's not the point of the electron transport chain. So as the electron moves down and energy is slowly released, that energy is going to be used to basically push or pump hydrogen ions from the mitochondrial matrix on the inside there to the intermembrane space. So it's going to pump it out. So out of the innermost part of the mitochondria out into the intermembrane space. And that's the whole point of your electron transport chain there. So slowly release the energy that we've trapped in these electron carriers and pump hydrogen ions out. All right, so that that step, electron transport chain, is going to help create our hydrogen gradient. So now we have more hydrogen ions outside in the intermembrane space than on the inside. And if you remember diffusion or osmosis, you'll remember that molecules want to move from an area of high concentration back to an area of lower concentration. So those hydrogen ions want to get back in to the mitochondrial matrix. They want to get back inside. The only way for them to get in is through a protein or an enzyme called ATP synthase, and that's this complex looking thing here. So in chemiosmosis, what we're going to see happen is that these hydrogen ions are going to start moving back through, and this ATP synthase here is like a little rotor. It's the smallest rotor that we know of in nature. And what happens is the hydrogen ions come in, they bind to the rotor part of the ATP synthase here. Remember when substrates and enzymes bind, remember induced fit, they change shape just a little bit. That change of shape causes the rotor to spin. If you follow just one hydrogen ion around, it goes one entire spin and then it gets released here. The spinning of the top part of the rotor here spins this internal rod here. The spinning of the internal rod causes catalytic sites down here in the knob to change shape, and that is going to allow ADP and inorganic phosphate to come in and actually bind together. So the movement of these hydrogen ions back into the inside of the mitochondrial matrix here is what's going to provide the energy to add phosphate onto ADP and create your ATP finally in the end. That's a lot. All right, so kind of looking at it all together here, we see the electron transport chain. So here's our inner membrane. So remember, you have the outer membrane of the mitochondria, inner membrane, and it's all going to go down across the inner membrane here. So we have our 4 protein complexes that pass the electrons through the electron transport chain. Remember the slow release of energy. That slow release of energy pumps your hydrogen ions out into the intermembrane space. They come back through the ATP synthase, and remember, it's a little mini rotor that spins, and that energy from the spinning is going to allow ADP, or provide energy for the work to create ADP and phosphate together to make ATP. Alright, so here's kind of a picture that sums it all up, and we see here's our stage 1 glycolysis, so we made 2 ATP. We have right here going on to make acetyl-CoA, that would be our pyruvate oxidation. We have our citric acid cycle, which is going to make 2 ATP. And then finally we have oxidative phosphorylation, which is your electron transport and chemiosmosis. Okay? And then you can see here, this is where you made the most. Okay, 32 to 34 ATP, so it's going to make quite a bit of ATP compared to the other. If we look at stage 1 or stage 3, stage 4 is where it really happened. And remember, the oxygen was the key. These other stages did not need oxygen. This final stage is where oxygen came in. And remember, it was the final electron acceptor at the very end of the electron transport chain. And without that oxygen, you can't have it go all the way to the end. So oxygen is very, very important. Without oxygen, basically the electron transport chain can't continue without the oxygen there. So you can't do oxidative phosphorylation. So in the absence of oxygen, someone was choking or someone drowned, lungs filled up with water, they're not getting enough oxygen in, and so they're only making a little bit of ATP versus a lot of ATP that they can make in the presence of oxygen. All right, last thing is going to be looking at macromolecules. So we know we can get energy out of carbohydrates because that was glucose, but I also told you we can get energy out of proteins and fats. These guys enter cellular respiration at different parts during different reactions across the 4 stages. So if we look here, proteins can enter here at the pyruvate stage, so they can be converted into pyruvate and enter right there. Fats can be broken apart into their main parts, which are glycerol and fatty acids, and that would enter here in glyceraldehyde 3-phosphate, and then continue through the steps just like we learned. They can also enter in through acetyl-CoA if we're talking about proteins. So at that step here, they can be converted into acetyl-CoA. Fatty acids, same thing, can be converted and enter at that stage right there. proteins once again. So proteins can enter at many points here within cellular respiration, can enter into the citric acid cycle. Okay, and then finally, of course, they'd go from wherever they entered, finally into oxidative phosphorylation and make a lot of ATP.